has asked me to rope in my services over there. So I may be, very decent.
वो better है, sir. Good morning, Dr. Amrish.
Very good morning, sir. Very good morning, sir.
Morning, Dr.
Always wanted to have you on our platform sometime and wanted to meet you, sir. It is very nice that we are meeting two most important dignitaries in pharmacy.
I'm to fry in front of Ranjit Singh.
नहीं सर ऐसे मत करो सर. पर ये बहुत अच्छा है, आपने प्लेटफॉर्म वर्चुअल है, हम एक्चुअली प्लेटफॉर्म पे मिलना ज्यादा अच्छा लगता है. फिजिकल प्लेटफॉर्म हो तो दैट इज वंडरफुल, बट स्टिल इट इज.
We'll have another one a big one in March around, so we'll surely be invited.
बिल्कुल.
Wonderful. I think there are 5 minutes to yet to go.
Yes, sir.
Yes, sir.
We should be joining a little later.
Yes, good morning, sir.
Good morning.
Good morning, all of you, sir.
अनम्यूट कर सकते हैं अपने आप को, मैंने म्यूट कर दिया था एक बार.
So we'll be commencing within 4 minutes.
तब तक आप लोग कुछ बात कर सकते हैं, डॉक्टर अमरीश, डॉक्टर अशोक.
Yes, sir. Sir, how is all going on at your end, sir?
All well. Best possible जो हो सकता है, we're trying.
Very nice, sir.
Every day improving, every hour improving.
Yeah. So SOP will be going for NBA inspection in January.
Okay, good.
So we have done that.
It is better to go for all these ranking and accreditation because you get so many improvements there.
Yes.
Some physical improvements, then you able to understand the system as such. अच्छा है. Carry on.
And this year we started M.Pharm in pharmaceutics and we started PharmD also, sir.
Okay, PharmD also. Great.
PharmD with 30 seats and M.Pharm 15 seats in pharmaceutics.
That is how you grow. More programs and you expand.
Yes.
That's good. आप कब से हैं शारदा में?
So it's been 1.5 years, sir.
Okay.
So before that I was in MD in Noida.
I know that. I know. आज कोई रिसोर्स पर्सन भी जुड़ेगा अभी नहीं?
Yes.
बहुत सारी हैं.
नहीं, वो तो सर तो हैं ही प्रोग्राम में ना, जो बाकी लोग हैं.
बाकी कल से सर।
स्पीकर्स जो हैं वो बाद में उसी दिन जुड़ेंगे, है ना?
Yes, sir, same.
Nasir has been my colleague in Bareilly.
Okay.
A very blissful morning to everyone. I, Dr. Ankita Tiwari, extend my heartfelt welcome to all of you on behalf of School of Pharmacy, Sharda University, to the 5 days International Faculty Development Program on the theme Cutting Edge Developments and Future Directions in Pharmaceutical and Health Sciences, organized by School of Pharmacy, Sharda University. We are deeply honored to have with us esteemed dignitaries who have made remarkable contributions to the field of pharmacy. I warmly welcome all our eminent dignitaries, academicians, delegates, and all the faculty members who have joined through this FDP. Now let us embark on this enriching journey of learning and collaboration, making the most of this opportunity to enhance our skills and knowledge together. Sharda University envisions to serve the society by being a global university of higher learning in pursuit of academic excellence, innovation, and nurturing entrepreneurship. It has 17,000 students from 95 plus countries, 29 states, and union territories, providing cultural diversity and students. It has 26,000 alumni who are today leaders in their realms. Sharda University is NAC A plus university with overall NIRF ranking of 86, and we stand 69th rank in pharmacy. School of Pharmacy was established in the year 2016 to offer an undergraduate pharmacy curriculum that is one of the most innovative in today's scenario. Our program emphasizes communication skills, problem solving ability, and a collaborative approach for providing patient focused healthcare. The innovative curriculum integrates foundational knowledge in the biomedical and pharmaceutical sciences with information and skills in the clinical, behavioral, and social sciences. This FDP will provide an overview of the latest advancements in pharmaceutical and health sciences, including the application of novel drug delivery systems, biotechnology, and digital health technologies. Let us begin this auspicious event by invoking the blessings of Goddess Saraswati, the embodiment of knowledge and wisdom.
या कुन्देन्दु तुषारहार धवला या शुभ्रवस्त्रावृता या वीणावरदण्डमण्डितकरा या श्वेतपद्मासना या ब्रह्माच्युतशङ्करप्रभृतिभिः देवैः सदा वन्दिता सा मां पातु सरस्वती भगवती निःशेषजाड्यापहा
I request Dean School of Pharmacy, Professor Amrish Chandra sir, to deliver the welcome address and officially inaugurate this FDP. Before I invite him, I would like to give a brief introduction. He is a very well-established academician, a research scientist, and a registered patent agent. He has experience of 21 years in academia and research. He has more than 100 publications, 5 patents, and 2 books to his credit. He has supervised many PhD scholars and PG students. And he has also a DST sponsored Indo-Hungary project with amount of 1.75 crores. So with this, I would like to invite respected Dean sir to please deliver the welcome address.
Thank you, Dr. Ankita, and thank you, Ranjit sir and Professor Whoop sir for kindly gracing the occasion. I would like to thank our Chancellor, Professor Dr. P.K. Sharma sir, and our Pro Chancellor Y.K. Gupta sir, and my heartiest thanks to our Honorable Vice Chancellor sir, Professor Dr. Sivaram Khara sir, for giving us this opportunity and this platform to be able to showcase this. And it is a great effort played by Dr. Nayyar, Dr. Ashok, and Dr. Rakesh in organizing this event and establishing all the contacts with such eminent speakers on today's occasion. And my thanks to Ankita ma'am, Bhumika ma'am for this. It is a great thing that we have around 1,000 participants currently with us who are joining us through our live link on YouTube. So that is one of the great things. So this we tried last year, and this year again we have done this, that we have a small core group on Google Meet, and it's being live streamed for a wider audience on YouTube. So with this, I thank everyone and I welcome everyone on this thing. When we talk of FDPs like cutting edge technologies in pharmacy and healthcare, it is very true that the pharmaceutical industry has been the cornerstone of healthcare, driving innovations that enhance the quality of life and improve patient outcome. Over the past decade, significant advances have been made in drug discovery, development, and drug delivery, propelled by emerging technologies like artificial intelligence, genomics, nanotechnology, and personalized medicines. So we have seen that these advancements are revolutionizing the way we understand the disease, the way we design treatments, the way we explore and discover new molecules and new identities, and also the way we manage new therapies. To talk of ChatGPT in a world of AI would be like talking just of the peak of the tip of the iceberg. A lot more lies beyond what ChatGPT is capable of. And if we talk of applications like ChatGPT, we can understand that we have yet not been able to fully exploit the potential that ChatGPT has. So the potential that artificial intelligence and machine learning has in pharma and medical field and healthcare sciences is something which is currently beyond imagination, and we don't even know what all it can do to healthcare. So these are transforming the pharmaceutical in all the possible ways, not only in drug discovery, but in value chain, supply, logistics, in all the fields. AI is making a tremendous amount of impact. Even in clinical trials, if we talk of AI, the AI can help in patient recruitment. It can help in real-time monitoring, analysis of clinical trial data. So all these tedious tasks, which are labor-intensive and human-intensive, can be delegated to AI in the today's world. Personalized medicine was and is and will always be there. We all know about the major technologies like CRISPR-Cas9 therapies, which are tailored to patient needs. These are very important things. And when we talk of further therapies, even CAR T cell therapies are very customized to patients' immune cell, which can provide very precision-based medication to the patients. We have always been talking of generic medicines and India being a leader in generic medicines, but the day is not far when India will also be a leader in biosimilars. Okay? And when it comes to biologicals, we had seen the way Pfizer, Biotechnic, and Moderna had delivered mRNA vaccines of COVID-19, and it was at an unprecedented speed and pace that such technologies were developed, such medicines were developed, and they were launched to the people at such an unprecedented speed. Biosimilars are cost-effective alternative biologicals, ensuring broader access to therapies compromise, and they deliver. They are very effective, as almost as effective as biologics. Nanoparticles, if we talk of nanotechnology, has again been a core. So we have seen nanotechnology doing wonders in cancer treatment, where they are able to target the cancer cells and leave out the rest of the cells. So Doxil is one of the medications, which is a liposomal based drug delivery system, which was launched and is doing great. When we come to further advances, we see that 3D technology also holds a very vast potential, which some of the speakers might be speaking on during this event. So, with the launch of the first tablet that is called Spritam, it is used for epilepsy. We have seen how 3D printing can evolve and what is the potential of 3D printing. So 3D printing can, at some point, it can be so tailor-made that it could be made for pediatrics, for geriatrics, or you can customize it even for at a patient level. How the regulations and other things will frame out during the development of 3D technology, that is one of the challenges that we need to see. When it comes to technology and future of pharma and healthcare, digital health will never be left behind, because with the advent of wearables like smartwatch, people can monitor their vital signs, they can detect abnormalities, they can track even treatment outcomes, and even the physician can be on the same page with the patient. So it is possible that the physician also knows what is going on in the patient's body through these wearable devices and all the things. So with this, I would like to say that the pharmaceutical industry is witnessing an unprecedented advances that are improving patient care, enhancing therapeutic efficacy, and addressing global healthcare challenges. From AI-driven drug discovery to nanotechnology-based drug delivery systems and sustainable manufacturing practices, these innovations are shaping the new era in healthcare. The future holds even greater promise, with ongoing research in gene editing, immunotherapy, precision medicines poised to tackle diseases once considered untreatable. And for this, we seek the collaboration, and we need to have a lot of brainstorming among scientists. And the core objective of this FDP is to make all the pharmacy professionals aware about what is going on in the pharma field and what all can be the future scope of work that can take place in the pharma industry. And with this, I inaugurate and I welcome everybody on this FDP. Thank you, sir.
Thank you very much, sir, for the welcome address. It is our distinct honor and privilege to welcome our chief guest, Professor Ranjit Singh, sir, who's a visionary leader and renowned expert in the field of pharmacy. We are truly fortunate to have him with us and grace this inaugural session of our faculty development program. Professor Ranjit Singh serves as the Vice Chancellor of Shobhit University, Gango, and he is the Director of the Adarsh Vijendra Institute of Pharmaceutical Sciences. A distinguished academic, he holds a B.Pharm, M.Pharm, and PhD in Pharmaceutical Sciences from Dr. Hari Singh Gaur Vishwavidyalaya, Sagar. He received more than 42 accolades, including gold medals for academic excellence, Distinguished Alumnus Award, and multiple Lifetime Achievement Awards, including Lifetime Achievement Award 2023 by Oma Welfare Trust and Dr. Sarvapalli Radhakrishnan Excellence Award in the year 2024. Professor Singh has over 3 decades of experience in academia, research, and the pharmaceutical industry. He has more than 210 publications to his credit in journals of national and international repute, 103 presentations, invited lectures, and 14 patent applications which have been published. He has more than 27 book chapters to his credit. Professor Singh is also a prominent speaker, having delivered numerous invited lectures and webinars. He has guided almost 38 M.Pharm dissertations and 31 research scholars for PhD degrees. His research focuses mainly on drug delivery systems, phytopharmaceuticals, drug standardizations, and with an expertise in oral drug delivery systems. His main area of research encompasses both novel as well as conventional oral drug delivery systems. He has held key leadership roles in several institutions and served as an expert on regulatory bodies like AICTE, PCI, NBA, and NAC. Sir, we are honored to have you here. I would like to invite for your talk. Over to you, sir.
Thank you very much. Such a nice introduction, encouraging. I will only say that I'm still a learner, and that process of learning and relearning, it is always on. You're never perfect. Thank you very much, Ankita, for such a nice introduction. And I could see from the program that some speakers are not there who were supposed to be there, including your Honorable Vice Chancellor. So just to start, Professor B.S. Bhoop, who is an authority in the field of pharmaceutical, everybody knows. And I am really thankful to the organizers who made me to interact and to listen to him today itself. Dr. Amrish Chandra, the Dean of School of Pharmacy and convener of this faculty development program on a very pertinent theme, cutting-edge development and future directions in the field of pharmaceutical sciences and health sciences. Professor Neher Parvez, Professor Ashok, Professor Rakesh, the organizing secretaries, the whole team, the conveners, the coordinators, the whole organizing committee, and invited guests, and those who are listening to us on YouTube and other such platforms, learned colleagues, friends, and dear participants. At the outset, let me convey my gratitude and heartfelt thanks to the organizing team, Dr. Amrish, Dr. Rakesh, who was in touch with me and who gave me this chance to be part of this inaugural ceremony. I am really honored and elated. This is an opportunity where I will be able to listen to Dr. Bhoop, who is acclaimed in the field of quality by design. We are in touch, but for some time, sir is not very active on social media, and yes, but he's there, he's available there. Let me congratulate you for thinking of such faculty development program. You have given it name International Faculty Development Program, possibly due to because the speakers are from international side. And I could see the program. I will just give one suggestion here that because there is a talk of 1 hour, I could see on from 18th, 19th, 20th, and 21st. There must be some interaction session. Just increase it so that question could be asked, and the faculty who are attending this, they are more wiser. When we see the current developments, a very good opening remarks by the Dean, who almost covered every aspect of what is happening in the field of pharmaceutical and health sciences. It has made my work quite easy. But, I will emphasize on one more thing. We are talking about health sciences and pharmaceutical sciences. I will speak something about health, because when we are healthy, only then we can perform. So our health is on priority. Professor Bhoop is the right choice for keynote address because he carries immense experience in the field of formulation development in specific and in pharmacy otherwise. He's globally acclaimed for his work on quality by design, based novel and nanostructured drug delivery and advanced pharmacokinetics. He has around more than 45 years of experience. I think the participants, their average age will come down of this. Served in Punjab University and is still connected to Punjab University. And it will be more appropriate if he's given more time to speak and share his experience and idea. Friends, we are all into a very fast-paced world. The happening around us is too fast. Technology is ever-evolving, we all agree to that, and so the field of health and pharmaceutical sciences. This is an era of technology. Particularly, we talk of disrupting technologies. They are all named by Professor Amrish: AI, ML, DL, AR, VR, MR, robotics, 3D printing, and all these. This decade, this third decade in particular of the 21st century, you will all agree to me that this is the decade of technology and skills. And we will be out of the scene if we are not skilling ourselves. So what I feel, skilling, upskilling and reskilling is the need of the hour. This message is for my young faculty colleagues who are listening to us right now, because this goes very well with what we call learn, unlearn, and relearn. If you are not upskilling yourself, then you will be out of the race. And the best thing is that you can skill yourself by many means, and one what I feel is important, self-learning. There are so many platforms which are available. You can choose the themes, you can choose the topics, you can go for short time kind of courses, certificate, diploma courses, where you can upskill, because it is not only the pharmaceutical sciences, but we have to think about the technology, how we can be technology savvy, and how we can be skilled in using the technology in what you call drug discovery and development, or patient care in health sciences or in pharmaceutical sciences as such. So updation and upskilling is quite essential for the faculty members. They must have knowledge about their subject, the technology, the skills which they need for effective delivery. And one thing I always realize and I emphasize, knowledge of rules and regulations is must. And once you know the rule and regulation, only then you will be able to implement them. We know the things happening around us, especially in the field of education, be it school education or higher education. We all know about National Education Policy 2020, which is in the stage of implementation, partially or completely. We all know about National Credit Framework, where the learning is being made learner-centric. We talk of this since decades, but this policy which is being implemented, it again emphasizes on student-centric learning, and it sees faculty as facilitator. Now the faculty is kept at the center, and it is our duty as faculty members that we deserve and become, such a person who are able to implement the provision of NEP. We are talking about outcome-based education, where we have to understand what we mean of all CO, POs, or learning objectives, high-order learning or other learning. We also know about ABC, then multiple entry, multiple exit system, internationalization of the education for Sharda University, because they have quite a big diversity, quite a good number of students from all around the globe. But internationalization means not to attract more students from other countries. So I will just call upon here that faculty member must upskill themselves. They must know about rules and regulations, and they must know about the technology. But I will emphasize one more thing here, that technology is important. It can be used for those tasks which are repetitive, which are mundane, but wherever the human intelligence is required, it cannot be replaced. So there is no fear that AI will replace humans, but yes, it will complement, and many good examples are given by Dr. Amrish, that how it is playing role in drug repurposing or drug discovery and development. It is able to collect huge data, analyzing it within no time, and giving us the way forward for new drug development. I will also talk on complete well-being. That is one area where we lack. So I will call upon to all my colleagues here, all my teachers colleagues here, that please go for self-care. अपना ध्यान रखें. What you call as complete well-being, when your health is okay, whether it is physical health, whether it is mental health, whether it is social, financial health, whether it is spiritual health, so many, or whether it is professional health. Your professional health will be good when you are knowledgeable. You will be knowledgeable when you will upskill yourself. So you can upskill yourself by self-learning, be self-motivated, and learn things from these own online platforms, learn things by networking, because with technology, people networking is important. You network, you learn. So self-learning, self-care If you follow them, it will lead to self-efficacy, where you will be successful all the way. You will be able to face all kind of situation, all kind of problems when we are talking about critical thinking or creative thinking or problem solving, but as mentioned in the opening address itself. So again, I will emphasize, please be healthy in all the directions. If you are healthy, you will be able to perform. As institution, we need to provide all the tools for these technological advancements. We need to provide techniques and skills to our fellow teachers so that they are able to use the technology to its best. Now, just on the cost of repetition again, I wish you all a good health. If you will be healthy, you will perform well, and I will be I'm more interested in listening to Professor Bhup. So I again convey my gratitude and thanks to the organizers for making me part of this great platform and giving me a chance to interact with the all the faculty members, young faculty members. And I wish a very grand and huge success to this faculty development programs. And I hope that all the participants will be more wiser when they will be able to attend and finish this program. Thank you very much. Jai Hind.
Thank you so much, sir, for inspiring us with your insightful words. And certainly the most important part which you told, like skilling, upskilling and reskilling is very important. So hope our faculties learn from that. Thank you so much. Today, we
Thank you very much, sir. Yeah, thank you very much, sir. And I would like to give a special mention to Dr. Satyendra. Sometimes what happens, the faculty members become so very close to you that you don't realize that you are actually two people. So it is Dr. Satyendra who has been working closely with me and organizing all this YouTube and everything that he has done. Yeah, Ankita ma'am, please continue.
Okay, sir. Today, we are privileged to hear from someone whose contributions have not only advanced the pharmaceutical education, research, and industry, but have redefined the possibilities for the future. It is a matter of great pride to invite our esteemed inaugural keynote speaker for today, Professor Bhupinder Singh Bhup, sir. Before I invite him, I know he needs no introduction, but I would like to take the opportunity to briefly introduce him. Professor Bhup is a globally acclaimed scientist with over 4 decades of contributions to pharmaceutical education, research, and industry. He has published more than 440 research articles, 16 books, and 92 book chapters for more than 16,000 citations and an H-index of 65. Recognized as a pioneer in pharma quality by design, that is QBD, he has delivered over 380 invited talks worldwide, training thousands of professionals in QBD and nanomedicine. With 35 PhD scholars, 11 postdoctoral fellows, 10 patents, and 3 technology transfers to his credit, Professor Bhoop's research has garnered over 7.3 crores in grants. And Stanford University has included him in the top 2% of scientists globally for 6 consecutive years, that is from 2019 to 2024. Professor Bhoop has received numerous accolades, including Lifetime Achievement Award 2024 and recognition for his outstanding contributions to the pharmaceutical industry and innovation. He has served in prestigious roles at Punjab University, including chairman, dean, and founder coordinator of national level centers of excellence. So we are honored to have you amongst us. With this, I would like to take the opportunity to invite Sir for his talk.
Welcome, sir.
Thank you so much. Am I audible?
Yes, sir, you're audible.
Thank you very much, Dr.
Yes, sir.
For doing the needful in a very, terse and appropriate and, excellent manner. And before that, I would like to thank Professor Ranjit Singh ji, the Vice Chancellor of the Shobit University, my great friend. I am proud and privileged to be associated with Professor Ranjit Singh ji. He's an able administrator and a dedicated academician and pharmaceutical researcher. We have been associated and we have been collaborating with each other for the doctoral guidance and the evaluation of the guided doctoral research works. And it was very, very pleasing to hear that he happens to be from Sagar, though he has been serving various universities with aplomb. But this is the acumen of all Sagarians that वो सागर को गागर में बंद कर देते हैं. That's what he did. That's what, he summarized the entire pharma profession and the health profession and emphasized on the concept of de-learning, learning and relearning and self-learning. And also, at the end, सागर को गागर में भरते हुए उन्होंने कहा कि it's the complete well-being which we should endeavor at and which should be the aim of everyone, be it the medical scientist, be it the paramedical scientist or the pharmaceutical scientist. So at the onset, let me thank the Dean of the School of Pharmacy, Dr. Amrish Chandra, and Dr. Rakesh Kumar Sindhu, who had been constantly in touch with me and persuading me to be part of this prestigious international EFDP. And of course, Dr. Ankita and many others who are behind the curtain, organizers of this very, event. I also sincerely wish that this event should come out to be a gala event, and it should serve the purpose, as Dr. Professor Ranjit ji was emphasizing. The purpose is that the participants, the young participants, I learned that more than 1,000 participants are connected with this EFDP through YouTube channel. I really compliment the organizers for letting the proceedings of this EFDP going beyond the precincts of Sharda University to national and international echelons. I really appreciate that. So the purpose is that all those participants are better informed about what's happening in the profession, in the society, and in the related health sciences. So I'm privileged to be talking about one aspect of the same. I wish to now use just a minute, new tab. I need to present. No.
Your screen is visible, sir.
Andy? Screen is visible?
Yeah.
Yet that first slide is visible.
No, sir, we are able to see the Google.
No, it's not that. It's not that. I should. I'm using Google Meet, after quite long period of time.
So in Google Meet, sir, if you unshare and you share it again, and if you can share the entire screen.
I'll do stop presenting, and then
share.
Share.
Entire screen. So there's an option called as present. Present now. Next to the smiley.
There's entire screen.
And then there will be a box which will be coming. You need to click that, and then the share button will be activated.
Yeah. Now, is it visible? No.
No, not now, sir.
Entire screen. It's okay.
So there will be a faded color screen that will be showing in that box. You need to click that first, sir.
Entire screen.
So when you click entire screen, a small box will appear, which will be a very hazy box.
Yeah.
That you need to click,
Okay.
and then the share button at the bottom will become activated.
A share button is active, but
You need to click share. Yes, sir. It will be coming. It is coming. Yes. Now you can go to the presentation, sir. Yes, your presentation is visible, sir.
is it visible now?
Yes, sir. It's visible. You can click the hide button over there.
Okay, that's it. Thank you so much. I am going.
Okay, so then please pin the presentation.
Yeah, is it visible?
Yes,
Continuous
sir.
paradigm.
Absolutely, sir.
Thank you so much, Dr. Amrish, for guiding me once again. So this is one practical demonstration of relearning. So I'm going to speak on one aspect of the entire universe which you are attempting to make the participants learn. That is the holistic and systematic development of drug delivery products using quality by design and other cutting edge paradigms. So I have structured my talk in this a little bit about drug development, including generics and the reference listed product, and what is the genesis of the same, and how do we arrived at how did we arrive at QBD and FBD, formulation by design? What are the models and designs available, and how can we apply them? In fact, let me share that for training the scientists, normally for industry scientists, I'm talking about the scientists who are employed in the industry, we normally take 1 to 1.5 days of session for training them. I have been privileged to train almost the scientists from thousands of scientists from almost 20 leading pharma companies. So the entire topic would be requiring much more time. But again, I'll try to follow the policy of President Jeet of trying to make include the sagar into gagar. So I'll try to make it as terse as possible. So drug product, as everybody knows, from technological perspective, it's a combination of so many agents. It's not merely API. It's containing so many excipients, functional and non-functional. I would call the high functionality or low functionality excipients. High functionality excipients include the drug release carriers, the surface active agents, the emulsifying agents, the suspending agents, the lipids, the polymers, and also the processes and low functionality excipients like the other additives, like diluents, etc. So where each one is contributing towards the product performance in their own characteristic manner. So needless to mention, the drug products could be branded ones or the generic ones. The branded ones are relatively costlier, and because of the patent issue, because of the proprietary issues, because the company that has discovered, that has pioneered the development of the particular brand has expended a lot of energy, time, and money on the same. While the generic products are the copycat products of the reference listed products or branded products. They are out to be similar in strength. They use the quality, the performance characteristics, and route of intake. And primarily it is bioavailability that should be matching with that of the reference listed product. The former U.S. President Barack Obama used to, emphasize the switch over from the branded products to generic products to make the drug treatment cost, more affordable in the US, and the same is applicable in other countries like ours too. So before I go in, let's feel a bit proud of our pharmaceutical scenario that our country is known as the generic capital and manufacturing hub to the outsource most of the drug products. It's a vaccine hub for WHO needs and even beyond. As Dr. Amrish was, accentuating, it's, an expanding biosimilar market too. And medical devices are being encouraged by the pharmaceutical and the governmental organizations to be again, making not only the country self-reliant but making other countries need, supportive too. And we lost a bit of API market to our neighboring country China, but we are re-establishing it. So the generic industry, we stand number 3 as far as drug product volume is concerned. But as far as drug product price is concerned, we stand 11th. Recently, in one of the talks by the DCGI, he emphasized that why should we concentrate on the products which are, highly sought after, so that we stand globally in, top 5 or top 7 in the drug product price as well. We are globally ranking 3 in volume, but 11th in price. But we should be, concerned about the profitability as well. So there are more than 3,000 companies. I'm sorry, and 11,000 manufacturing units. Every third pill taken across the world is from India, and 40% generic needs in the US is from met in India. More than 65% WHO vaccine needs is from India, and total 70% vaccine needs are made by India. Every third drug master's file filed in USA is from India. One of the largest pool of scientists India has got, and the US 25 billion market, which is growing at a phenomenal rate of 10 to 20%. Year over year, it keeps varying. Last year it was 11%. So a drug delivery system is a formulation with some special characteristics. It's a programmed drug release. It could be used for controlled release, fast release, or targeted release. Where the time has been tempered with, it's called as temporal. Where the space has been tempered with, that's the target has been achieved. Targeting has been achieved. It's called as spatial type of delivery systems. And nanostructured delivery systems, needless to mention, constitute a chunk of those novel drug delivery systems, which includes the lipidic and the polymeric and the lipid polymeric systems and the carbon-based systems. Important ones and include SNEDs, solid lipid nanoparticles, nanostructured lipidic carriers, nanomicelles, liposomes, nanoliposomes, or when they are in the nano limits, nanometric limits. So they are called as nanoliposomes, nanolipopolyplexes, quantum dots, dendrimers, graphenes, carbon nanotubes, fullerenes, so on and so forth. Now, be it the conventional drug products or the novel drug delivery products, the world has been witnessing large number of product recalls and rejects. As I had this 2016 statistics only, 7-8 years old, but the scenario still remains the same. So the number of recalls are exponentially increasing. And these are not limited to prescription drugs. These are extended to over-the-counter cosmetics, medical devices, dietary supplements as well. And the US FDA, has been, emphasizing that these recall scenario. This is June 2023, a poster from the US FDA. that it uncovers failures in Indian pharma factories as well. So this brings bad name, because the cause is ultimately poor quality, consistency, and integrity. So how can quality be assured? The question arises. So the obvious answer is by reducing variability, that is inconsistency, through rational and systematic development and optimization. So, my dear young friends, most of you are young by age or young by heart, either way. So, if you look around, this is a typical process where a coated tablet is being prepared, and you look around. The process starts with the granulation, then drying, and co-milling in between, and then mixing, then rotary compression, then coating, then imprinting, and finally, a product which was, here, yellow colored product, gets into the form of imprinted tablets. So each of these processes are marked with some kind of variability. So, my dear friends, to organize this and to optimize this variability leads to you to the products with the desirable product quality. And this is a typical cause and effect Ishikawa fishbone diagram, which is quite typical in the management practices, in the industrial science practices. If you look around, here it's shown that not only the materials, that's the APIs and the excipients, it's the men, that's the
हाँ जी, अब आ गया।
Hello. हाँ सर, कैसे चला गया?
पता नहीं सर, वो अपने आप ही लैपटॉप हो मेरे वाला।
प्रोफेसर की साइड से ही हुआ सर।
ठीक है, ठीक ठीक है बस।
There's a technical glitch and we'll be resuming shortly. Just for the information of the participants, the link of the feedback will be shared at the end of the FDP, day 5.
Am I audible, sir? Please unmute your.
Am I audible?
Yes, sir. Yes, sir. Very much audible, sir.
Okay, I'm just entire screen. Okay. Okay.
Okay, sure.
Is it visible?
Yes, sir. It is coming. Sorry.
Yeah.
Yeah. So we are on the first slide, sir, right now.
Okay, no, I'm changing. Yeah. Oh, sorry. Yeah, I should have stopped sharing. Sorry for the technical.
No issues. No issues, sir.
Now, is it visible?
Yes, sir, it's visible.
Yes, sir.
Now we are at the same point where we left, Anna.
Yes, sir.
Okay. So, question arises whether we had not been optimizing our drug delivery systems, we had been adopting the approach which itself is suboptimal. That's one variable at a time or one factor at a time, or also called as cost or short-term. So it had so many disadvantages. So time does not permit me to go into the all the disadvantages, but I have highlighted a few of them. That is, this OFAT approach was quite strenuous. That's effort consuming, quite uneconomical. That's quite time consuming. It was not suitable to find errors, and needless to mention, it was not suitable to plug errors as well. If the diagnosis of any problem is not good, how can the treatment which is following the diagnosis be good? So we were unable to find the errors, so we were unable to plug the errors as well in the OFAT approach. It results only in just satisfactory solutions. Detailed study of all the variables is prohibited. You know? And there is no cause and effect relationship in the development of during the development of the formulation system. So it was just like blind men, the old adage of so many blind men and an elephant theory. So blind men, had encountered the feel of a big animal, that's elephant. So they tried to explain it to each other by feeling a part of it. One person told that it is just like python, the one who was feeling the trunk part. Another felt it was like a tree trunk. Another felt it's like a snake which was feeling the tail part of it. So everybody had different notions. The animal remained unexplained because they were not able to see. So that was currently the scenario, in case of the shotgun approach or OVAT or OFAT approach. So we were not able to define the formulation systems per se. But fortunately, we had another approach which we had been ignoring otherwise. So this is called as design of experiments, which is a rational and systematic approach that enables teams to learn about product and process behavior by running a series of experiments such that you get maximal information using minimal experimentation. I repeat, maximal information, minimal experimentation. This is the technique which pharmaceutical industry had not been adopting, while other industries like automobile industries, etc., had been regularly using to their fullest advantage. Needless to mention, DOE is a systematized approach, and it had so many advantages. Again, time does not permit me, so I need to highlight a few. It requires fewer experiments. I repeat, maximal information, minimum experimentation. So experiments required are much less. And it yields the best solution, the best, I reiterate, the best solution in the presence of possible competing objectives. And you can trace the problem and you can plug the problem, that's rectify the problem, much in a much more easier manner. It leads to comprehensive understanding of the formulation system. That's what FDA and other global regulatory agencies are today accentuating, that we need to have product and process understanding. I keep telling my students that if a doctor commits the mistake, it is the life of that particular patient who's getting treated under in danger. While if a pharmaceutical scientist who is serving the industry and preparing formulations, drug formulations, commits a grievous mistake, it is the lives of hundreds of thousands of the patients who are consuming his products in danger. So, I leave it to the imagination of the listeners, whose job is more responsible. So that's why FDA, EMEA, TGA, and other agencies, regulatory agencies, had been emphasizing, even WHO had been emphasizing that there should be comprehensive product and process understanding so that such type of errors are prevented from actually occurring. So this is possible using DOE, and you can simulate the product or process behavior using model set of equations. And above all, resources are very, very, useful these days, are becoming much more, useful, much more limited as well, like developmental time, developmental effort, the API and other materials, and the overall money. All these can be saved significantly if we use this scientific and systematic technique, DOE. So, my dear friends, so the bout is between OFAT technique and versus DOE technique. Like this FAT and SMART, wrestler working with each other. Let's see who wins the bout. So we carried out in 2005, around 20 years, 2 decades back, one experiment on drug delivery systems. That was on tenoxicam hydrogel for transdermal formulation, transdermal application, and we used two, final functional excipients, that is gelling polymer, that's carbapol ETT 2000, and penetration enhancer, that's oleic acid. and we used both the techniques, OFAT as well as DOE. Look, in the first case, flux, that is flux through human cadaver skin, was plotted versus gelling polymer concentration, and the second point gave maximum flux, and similarly penetration enhancer concentration was plotted versus flux, and the point in between, third from the right, gave the maximum flux, and we made a formulation with the gelling polymer concentration and penetration enhancer concentration giving maximum flux, and then the point of intersection of the red beads in the lower diagram, came out with the OFAT or OVAT, or cost optimum. While we carried out another experiment using DOE, and it yielded the truer optimum, that's the black spot, which is way different from that of the optimum which was found using the OFAT, OVAT, or cost approaches. So the results are the results can be phenomenally different, as in this case. So another way, factor A is optimized, and at the same settings, if another factor is optimized, you get apparent optimum, while the real optimum is missed altogether. Various scientists had been telling in a different manners. While I keep quoting Potzak as gambling, like guaranteed that the formulation which you'll be getting using OFAT or OVAT will be really optimum formulation. While using DOE, you'll always get the first class formulation which can never be challenged. It will be the best under the competing objectives. So the pharma world, got shaken in 2003 when the popular magazine, US magazine, the Wall Street Journal, a weekly magazine, which almost half of the US and almost one third of the developed world reads every week, gave this, headlines in their one of the weekly journals, that the pharmaceutical industry has a little secret. Even as it invents futuristic new drugs, its manufacturing techniques lag far behind those of the potato chip and laundry soap makers. It was a big blow to the pharmaceutical industry, including the pharmaceutical regulatory bodies like FDA, etc. So, because they have all been challenged, that the techniques which have been used in the most sophisticated kind of, supposedly most sophisticated kind of products, are very outdated. While the industries like pizza industry, dental implants, nut and bolt, potato chip, and laundry soap makers, and oil exploration industries have been using such type of techniques. So US FDA, got awakened. So they brought in due course of time the guidelines of quality by design, of which DOE was almost an essential component. So quality by design, they explained as a systematic approach to design product and process to meet the patient needs. So patient needs is servoprior. It's the most important over here, at the desired quality levels without relying upon periodic inspection and testing of quality. So the purpose of QBD was that the quality should be built into the system, rather than quality should be ensured just by inspection and testing. If it meets, it's okay. If it doesn't meet, it needs to be discarded. It's not a hit and trial. It has to be systematic effort. So this lady, till about 10 years ago or 8 years ago, when Trump took over in his initial phase, she was reigning the FDA's scene, and she was a very familiar face who defined pharmaceutical quality as the function of drug substance, that's API, excipients, process, packaging. And had I been involved, I would have suggested, and or others, QBD scientists been involved, they would have suggested that add interactions along with. So pharmaceutical quality is not only a function of drug substance, excipients, process, packaging, and interactions have to be taken care of. The services of the father of QBD, that's J.M. Duran, were called, who had shifted from US, born and brought up in US. He had shifted. He was not listened to in US, so he shifted to Japan, where he led successfully several automobile industries like Toyota to produce, their cars, their automobiles, having the quality in a systematized manner using his quality, paradigms, and which were later named as quality by design. So the father of QBD concept is J.M. Duran. So his services were called, and he was not willing to come back because he told that nobody paid heed to me when I was there in US. So he was assured that his policies would be implemented in toto now in the pharmaceutical sector. So he came back, he suggested, various meetings had happened, and QBD concept was promulgated, and there was no looking back. In 2013 January, all generic products which were to be entering the US system had to be QBD compliant. So this Duran's philosophy of QBD was quality by planning, not merely by chance. Quality by design means quality by planning. So what is quality? What is design? How does design deliver quality? Quality, my dear friends, I looked into when I made my first presentation. I think that was in the year 1995 at Cadila Pharma, Ahmedabad. So I looked into the word quality, and dictionary told building excellence in any trait. And I looked into excellence, so it gave building quality in any trait. So, my dear friends, quality and excellence, I personally feel, are synonyms. So, and quality is not static. The definition of quality of any product which was stated 20 years back does not hold good. So it has to be dynamic. So it has to change with time. It has to change with the scenario. It has to be updated every now and then. So design, what is design? Some people, rather most people, take design as the experimental design, a statistical design. No, design over here is the planned experiment. In day-to-day life, we also say, this guy doesn't seem to have nice designs. Means iske irade theek nahi lagte. Nice designs. Plan. Irade means plans, and irade means designs. So design means planning. Design means your bent of mind, your approach. So design is systematic approach to attain quality by requisite planning. We defined it in 2011, and Swiss Pharma gave the definition of design as it's a conscious intent. It's your intent. It's your intention. It's your plan to deliver a product or process with a predefined quality. So what is QBD all about? It is the rational mindset of doing things right and to follow the process. Not only that, it's the amalgamation of scientific principles to that rational mindset. Not only that, it is the adoption of those scientific principles and rational mindset to obtain products which are scientifically deliverable, which are scientifically validatable, and which are scientifically reproducible, and which do come across all the, which do satisfy all the needs of the quality, integrity, safety, efficacy, and other pharmacopoeia and regulatory requirements. So doing it right from the start is the QBD aim. And it's all about patience, you have to understand the patient needs, you have to design and develop products meeting those patient needs, and you have to design and develop the manufacturing processes capable of delivering the products and products meeting those patient needs. So QBD settings let industry even go beyond the requirements which are normally, anticipated. You just look around, it lower specification limit and upper specification limits. That's the normal customer expectations, but it can modulate even beyond. QBD has the power to let it below lower specification limit and satisfy your product even above specification limits. So this is out of the FDA side, which was, initiated in 2011-12 to prompt the industry to adopt QBD guidelines. So the current state and the next state, the cost would be higher, but the desired state will be economically much more affordable. Phenomenal cost implemented. So why to implement it? I had been sharing with the pharmaceutical industry that there will be good return on investment, ROI, and knowledge sharing would be much better. Improved time to market. They understand this concept, time to market. In generic industry, whosoever launches the generic, rules over the scene. So improved time to market, and cost would be low because there will be less of recalls, less of rejects. Cost will be automatically lower, and people are skeptical about generics. If I have to buy, product, my daughter is here, and daughter says, my her son, that's my grandson, is unhealthy, and please go and fetch expectorant syrup from the market. I'll go to buy the expectorant syrup from the market. And if the pharmacist tells me that there are two versions, the generic and the branded is costing, say, 200 rupees, and the generic is costing around 30 rupees. Which one would you like to buy? I would search my pockets, and if I have got 200 rupees note, I will buy that the branded ones, because I do not wish my young one, my grandson, to have some kind of an ambiguity, because I'm skeptical about the quality of the generics. So that's skepticism, that doubt about the generic quality would go if we adopt systematic approaches like quality by design, and if we propagate that we have been adopting such type of systematic approaches. There will be minimal post-approval changes, and the products will be, I have already emphasized, Obama emphasized, the approach will be more, cheaper, affordable. And QBD also, provides you the flexibility. Time and again, the industrial scientists had been asking that, the regulatory framework does not provide us with the flexibility. They provide us a very fixed approach, So QBD tends to give you the desired requisite flexibility, in the sense that I'll be discussing design space. You have to move within the design space. So there is enough possibility of moving within that design space. So the flexibility is also provided in the basic concept. So I keep telling the pharmaceutical industry that it's neither the strongest of the species nor the most intelligent ones, but those who are most adaptable, those who are most responsive to change, they will survive. So in pharmaceutical industry in India too, I'll almost 80 to 85% industries, uh, responded to that QBD change and made a benefit, and some 10-15%, lagged behind others only because of the delayed learning and implementing of the concept, and they missed the chance to export their produce in the US and subsequently in the European and Australian and the British environments. So, my dear friends, it's not QBD is not merely useful for industry and the patients. It's useful for regulatory agencies as well. Look at 2008 filing on the left side. How voluminous it used to be. Lot of documentation was required, and post QBD implementation 2014 filing. The volume is around 1/5 or 1/6 in comparison to the previous ones. So pharma quality excellence, and it needs to be allured using federal QBD compliance. So FDA gives QBD as the allurement, and leads the pharmaceutical industry towards excellence. So compliance is subsequent. Excellence precedes compliance. If the product is excellent, compliance would automatically be there. That's what FDA wants, every product to be QBD compliant. If it is QBD compliant, it will be federally, regulatorily compliant as well. Why QBD implementation reduces post-approval submission load too. Earlier, it used to be data rich but information poor. Now, you can provide any kind of data using the computational means, using the means which one has got. So quality by testing, the other approach, wherein every process was like black box. You see over here, every process. So testing was required at every stage. Inspection was required at every stage, and end testing was mandatory. While quality by design, so testing is not required at every stage, and end testing is also not required. But end testing is carried out invariably, just as a practice to validate that whatever had to be achieved has been achieved. So a traditional QBT approach and quality by testing approach, where testing used to outweigh the design, the planning part, while QBD approach, testing and design tend to balance each other. And QBT, that the earlier approach is just like the chaotic traffic of New Delhi. You have seen it. And QBD, on the other hand, is traffic regulated on Los Angeles, which I visited a couple of times, in the last 2 decades, And QBT is like diving in the ocean full of uncertainty, quality by testing. And quality by design is like driving on the well-carved path on the same ocean. QBT is like the tip of the iceberg, while QBD is the entire iceberg, including that tip. So QBT is like trying to fit one attire on all sizes, but QBD lets it happen. QBT banks upon knowledge exchange from one person to another, one team to another, one company to another, while QBD banks upon knowledge integration, not exchange. All sorts of departments tend to sit together, integrate together their knowledge, and what eventually comes out will be an immaculate product. So QBD is in tandem with QBR. QBR is question-based reviews. Today, the products in FDA are allowed and approved only if the scientific team is able to answer to the queries. So that's what many of the FDA reviewers shared with me, during some conferences as well, when I was also learning the details of what FDA want under QBD from the industry. So that's what they tried to explain, that whenever they used to ask the scientists from India, from China, or from Brazil, which used to export their products to US, say, how did you make this product? They used to tell, sir, not only you are not understanding, so this product is, having similarity not only in the dissolution, disintegration, in friability, but it tastes too, in color too. Well, that's okay. How did you make it? And there used to be a big vacuum, big, flaw. So because they were unable to explain, because we people are very good at copying, but or reverse engineering, but we are not. We had not been adopting the scientific and risk-based strategies. So we were not able to perform reasonably well in question-based review. But with QBD in place, our scientists are able to defend question-based review as well, because they understand their products and the processes much better. So the FDA timeline, I have been telling that January 2013, they asked every product to be compliant with the QBD practices. But January 2013 is more than 1 decade old. Situation is still not complete. It's still alarming. So these are the salient steps involved in QBD. First is quality target, product profile. That is, you have to have some objective. That's QTPP, it's called as. And for that, you need to identify some critical quality attributes. This is taken out from the QTPP itself. And you tend to embark upon some input parameters, which may be critical material attributes and critical process parameters, which need to be linked to the critical quality attributes. And that enables the industrial scientist to develop a design space. I'll touch upon that aspect a little better. And the entire process from QTPP to design space is now recorded as a strategy called as control strategy. Implementation of that control strategy leads you to continuous improvement of your product, batch after batch, day after day, month after month. You can continuously improve your product over the previous batches as well, while remaining within the design space. So QTPP is the prospective and dynamic summary, like what is route of administration, what is dosage strength, what is container closure system. Say if it is a drug delivery product, what is drug release, and of course, besides that, the conventional requirements of sterility, stability, purity, and drug delivery attributes like aerodynamics for the inhalers and dissolution for oral products are taken up as the QTPP. Say, for example, this is a tablet dosage form example, where QTPP is the dosage form, route of administration, strength, tablet weight, appearance, identity, assay, impurities, etc. And out of that, some important characteristics have been borrowed after team, discussions, after the meeting of the stakeholders and embarking upon the three parameters out of QTPP, which are reflective of clinical efficacy. That's assay, content uniformity, and dissolution. Assay, that's very important, to ensure that the labeled amount has been, assayed, has been the content has been found. Dissolution, whether it gets released in due course of time or not, and content uniformity, that is to ensure the consistency, that's homogeneity, and for safety impurity levels. So these are the CQAs have been identified from the possible QTPP for a tablet dosage form. This is how it's done. So, critical quality attributes, these could be physical, chemical, biological, microbiological property of the dosage form that should be within certain range. And how are CQA selected? What are the parameters? What are the criteria? These should be selected on the basis of profit of the company? No. Operator safety? No. Environmental safety? No. Only for patient benefits. So CQAs are selected on the basis of the patient benefits, patient efficacy, patient safety, patient quality, patient acceptability. If those are the criteria on the basis of which CQAs are identified. And CMAs and CPPs. CMAs are critical material attributes of the products, that is API, excipients, etc. CPPs are the process factors like granulation time, granulator speed, etc., etc. So this is again out of the FDA site. You just look around, there are two semicircles. So semicircles are contributing towards product and process understanding. So last one is product and process understanding. So that's what the endeavor and the aim of the and development of any product under the QBD perspectives has been. So what are the guidelines available to guide us on QBD? It started with the, in fact, Q6, ICH Q6 guidelines, but later on for Q8, in first time, the quality by design was defined in ICH Q6, but that was in very primitive fashion. Q8 was for pharma product development. Q9 was for quality risk management. Q10 was quality systems. Q11 was for development of API, drug substances. Q12 was for life cycle management, because QBD is not mainly for product development or API development, it's for the entire product life cycle. And Q13, which have been promulgated relatively recently in 22 and 23, continuous manufacturing, and Q14 on systematic analytical development. I'll touch upon these as well. And plus PAT, that's process analytical technology, PAT guidelines, which are complementing with that of the QBD. So quality guidance Q12 to Q14, now Q12, Q14, Q8 to Q14 have been adopted by not only ICH itself, by FDA, WHO, EMEA, that's Europe, MCC, that, and Health Canada, SFDA, MHRA, that's UK, PMDA, that's Japan, Australian government, that's TGA, and MCC like South Africa. So all SFDA, that's China. Even in India, the revised Schedule M has many provisions of quality risk management and a bit of the CQAs and CMAs requirement. And so this has to be not entirely QBD, because our MSMEs are not coming up to the mark as far as the quality guidelines are concerned, and imposition of QBD would have further burden them. So only part of it, that's quality risk management, has been implemented. So when QBD has been the talk of the town, in 2012 we proposed another term, that is formulation by design. Formulation by design is specifically related to formulation development. So not only it contains the state of art art and science of drug product development. So, drug product development is not merely science; it's art as well. So, taste, organoleptic characteristics, etc., they come under art. Drug development, drug product development, QBD enabled understanding. It requires DOE knowledge. It requires quality risk management. I just touched upon that. So we were the first to, introduce this term in uh, 2010. And although in expert opinion in drug delivery, there was a lot of having our paper been accepted. The editor wrote so let it be quality by design. The term should be quality by design for formulation. Like, no, nothing doing. We wish to introduce this term, formulation by design, and believe it. Your journal would be considered as a pioneer in helping this term becoming a pioneer, becoming in place. Now today, FBD is very common term adopted in pharmaceutical industry in by consultant, and this is for Delta company using formulation by design. So formulation by design, if you look around, has become a commonplace dedicated to formulation, application of QBD in formulation development, systematic formulation development. It's the best tool to match innovative products performance. So earlier OFAT approach used to hit here and there, while the QBD guidelines on FBD always hits the bull's eye. It makes the best possible formulation. So OFAT had many disadvantages, while FBD is systematic, robust, holistic, yields the best solution. So a drug product is in FBD is considered as a system. It's considered as a transfer function where X is transferred to become Y. X are the inputs, raw materials, processes, and Y is the product. And there are other variables which are called as uncontrollable variables or nuisance variables or noise variables, the humidity, the temperature, the dust around, etc. So U is not controlled, but X is controlled and Y is estimated. If Y is perfectly estimable using X, it's called as a white box. If Y is not at all estimable from the values of X, it's called as a black box. But if it is moderately estimable, it's called as a gray box system. While using OFAT, it used to be invariably black box or nearly black box status. Predictability was very poor. But using FPD, it is always a white box or grayish white box status, very high predictability of Y with respect to X, it can be obtained using FPD. So it tells you what's happening inside. So there are some technical terms which need to be understood. That's variability factors, design, software, model diagnostics. Time does not permit me, but those who are seriously interested, can contact Dr. Avinash and Dr. Rakesh Sindhu. So I'll provide them with enough materials. Some of the articles which have now 500 or 450 citations, and on DOE and FBD, are highly sought after. The journal, markets them at very high exorbitant prices of $200, $250. But those comprehensive articles are available with me. I being the principal author, corresponding author, so I can supply those articles to Dr. Rakesh Sindhu and Dr. Amrish for your usage. So what is objective? Otherwise, it's called as QTPP. Variables could be CQAs, CMAs, CPPs. CQAs are dependent. CMAs or CPPs are independent variables. Levels are the values assigned to these CMAs and CPPs, which are also called as factors. CQAs are also called as responses. Experimental runs or trials are the studies carried out. Factor space is the, say, for example, had this exercise been carried out in as the not online but offline, FDP. So you would have thronged to Noida, and Noida would have been explorable space, and within that, Sharda University would have been the knowledge space, and within that, this school of pharmacy would have been the design space, and within that, the auditorium where this conference is being, this FTP is supposed to be held would be called as the control space. So these spaces are being used, while the design space is the most relevant amongst these. So variables are CQAs, CMAs, and CPPs. CQAs are dependent variables. CMAs and CPPs are independent variables. So we carried out one exercise way back in 2006. So while preparing quaternary system, that's super saturated systems, or also called as solid dispersions. So we had two carriers, Polexamer 407 and PVPK25. Polexamer 407 was changed finally between 25 and 75 milligram as the low level, high level, respectively, while 50 milligram was called as intermediate level. While PVP K25 was varied between 50 to 100 milligram, 75 milligram being called as the intermediate level. And interaction is when the response variable, that CQA, is plotted versus CMA. When at low and high level there is a parallelism, it's called as no interaction. When there is divergence, it's called as synergism. When there is convergence or cross sectioning, it's called as antagonism. Who knows it better than we people? What is synergy? What is antagonism? When two substances are having more than the anticipated effect, it's called as synergism. When antagonism is the inverse of the same. And response surface plot, which is 3D plot between two independent parameters, and that is tween AT and completol versus the CQA, that's particle size, over here. So if you look around, it's a three-dimensional kind of plot. So and if you look at the top view of the same, so the top view would be looking like these curved lines, and with the tween AT and completol, as the on the axis, while the CQA here it is entrapment efficiency on the other hand. So the lines over here, each contour line, these curved lines are called as contour lines. It could be curved, it could be straight lines. In this case, it's curved line. Indicate the innermost line indicates 88.1358 is the value encountered over this curved line, and 82. So this is contour plot is the plot containing contour lines where the independent parameters tween AT and completol need to vary in such a way that the CQA, that's entrapment efficiency, remains constant. All these are the outputs of normally a software, software made out of FPD. So there are 5 steps in journal, my dear friends. Again, time that does not permit me to go into the details. In the first step, you need to define QTPP and CQAs. In the second step, you need to prioritize on defining CMAs and CPPs, and the techniques which are used are called as factor screening and risk management. In the step 3, we need to carry out experimentation as guided by experimental design, and an analysis needs to follow. In the step 4, we need to modelize and validate those that data analysis which we have carried out under step 3, and embark upon design spaces and control spaces. Final step, that's fifth step, that's pilot plant scale up production and commercialization, is carried out in the industrial environments. So QTPP and CQAs I've already defined. Step 2, CM and CP's prioritization. You need to find out the parameters with the highest priority. So it's based upon the Pareto principle of factor sparsity. That's 80% of the effects come from only 20% of the causes. So, for example, 80% of the wealth of the world is owned by 20% of the rich. Similar is true with respect to India. But some people, put it like this: that it's 90% of the wealth of India is owned by 10% of the rich. Whatever, it's subject to verification. Pareto principle is based upon 80-20 principle. Even those who join marketing industry, they are told that 80% of the consumers tend to consume 20% of your products. You need to concentrate on those 20% of the consumers who tend to utilize 80% of your products. So we need to not squander our energies and time and effort on rest of the 80% of the consumers who just account for 20% of the products. So 80-20 principle of factor sparsity is applicable over here. So we need to concentrate on 20% of the variables which are accounting for 80% of the dosage form properties. So, and this is called as factor screening, and together with the quality risk management. So many material attributes, and after carrying out these exercises of factor screening and risk management, you are left with vital few, that's critical material attributes and critical process parameters. Now, this slide tells you how screening works. It's just like separating rice from rice husk. So rice over here provides simile to the factors, while the husk is with respect to the unwanted variables. So it's 20% of the influential variables are called as factors. So we need to concentrate on these factors. So please, let's focus on this slide a bit better. So if the input variable is a factor, that's influentiable factor, and it is finally identified by the scientist and regulated and optimized by the scientist. So the outcome will be a true optimum formulation. But if it is a factor, you as scientists are unable to identify the same. God is the savior. The outcome will never be, accurate. If it is not a factor, but you have unnecessarily identified the same, nothing to worry about. But of course, you have squandered a lot of energies, time, and effort. But if it's not a factor, you have not been able to identify no effect because it was not a factor, you have not been able to identify. So, my dear young scientists, a factor, influential factor, must be identified to arrive at the true optimum formulation. Even if it's not, a not influential factor is also identified, it may not lead to the imperfect formulation. It may lead to squandering of time, But most important is that a factor has to be identified as a factor. So how is it identified? Again, dedicated computer software comes to your rescue. So this is out of the output of design expert software. You see, the points which are falling on the line, that's in the beginning of the line, they are falling the line. So it's like a flock of sheep. While the points which are encircled and which are away from the line, that's B, C, and BC, they are away from the line. They are not away, they are not part of the flock of the sheep. जैसे कबीर used to tell जे पाछे पंडित गए पाछे परी बहीर एक अवगढ़ घाटी राम की तह चढ़ रहे हो कबीर. So it's not following the flock of sheep, it's having their own path, steering their own path. So BC and BC is together is interaction. B is another one parameter, independent parameter. C is another parameter, and BC is the interaction between B and C. So they are the influential factors, while the lines, the points lying on the line, they are having negligible effects. These can be ignored, while B and C are most important influential factors. How is it being revealed? This is called as half normal plot, which is one of the very important tool of the software design expert. So another is Pareto chart. So Pareto chart also gives you the magnitude, the height of the chart. So if the height of the chart is exceeding Bonferroni limit, here it is, the upper red line is Bonferroni limit. So there is absolute no ambiguity that it's going to be influential and highly influential. You cannot afford to ignore this. But if it is above t value limit, which is that dark blue limit, so then two, it is influential. So over here, C is influential, A is influential, and AC, that is the interaction between E and C, are influential. While others, which are lying below the t value limit, this is from the actual data, are non-influential. They must not be ignored. They must not be taken into consideration while optimizing the formulation. While optimizing the formulation, only the prioritized variable A, C, and the interaction between the two should be optimized. So risk assessment is another exercise which is carried out in industrial milieu. That's what I was sharing. Quality risk management has been made essential even in the device schedule M, which is to be implemented, in all the pharmaceutical industries. Particularly, it's a big challenge for MSMEs. The big pharma companies are already implementing, but the upcoming MSMEs, need to implement this as a routine matter. So it's quality risks allow one to walk on the tightrope among possible risks, possible dangers. Not only that, it trains the scientists to walk on the tightrope, but encountering minimal dangers. It provides a cushioning effect as well. So to make it more interesting and illustrative, so over here, you look around, the guy is feeding the nutrition, the meat stuff to the lion within the cage. Isn't it, a risky process? He himself is inviting the dangers, along with the diet which has been, presented to the animal, the canine animal. So it's extremely high. So next time he, covers himself and provides, the same kind of diet. So the risk is reduced. So over here, I have tried to. So the seriousness is 10, and the likelihood of attack is 10. Over here, likelihood of attack remains 10, but the seriousness is 80% because the damage to the individual will be less. And the third is risk reduction. He does not come in direct contact with the ferocious animal. So the likelihood of attack is 60%. If the animal has been watching, no sooner the person comes, he'll attack. So the possibility is 60%. And possibility of the damage is also 60%. While there is another way, of risk minimization is, putting the food in place and then opening the slot. So there's no seriousness. There's no contact. The risk is minimized. So this is called as quality risk management. This is risk minimized. Risk, I got risk maximization, and this is risk reduction, and then this minimization. So all these processes happen to be applied in pharmaceutical practices too, and there are approaches for the same. That's comparison metrics, risk estimation metrics. I'm bypassing rest of these, and failure mode effect analysis. Three important ones: comparison metrics, risk estimation metrics, and FMEA. That's failure mode effect analysis. So overall, the low risk variables are always to be ignored. The high level risk variables are always to be given high priority in the during the optimization, because they are likely to play havoc with the efficacy and or the safety of the formulation. While medium, you can take a call on the same when you sit as a team. So this is a typical risk estimation table. You see, high three out of the 9 possibilities, 3 are with high level of risk. So at these points, the variables need to not be ignored. In any case, while the 3 points which are green ones, they can be simply ignored, while for the yellow ones you can take a call. Another way of telling, probability, here it is severity versus likelihood, here it is severity versus probability of occurrence or likelihood. They are the same. So in the last technique which I promulgated in the previous slide, that is FMEA. FMEA. So during FMEA, there are one more parameter, one more criterion taken into consideration. That's called as detectability. So the first is to detect possible failure modes. When can a particular formulation fail? Failure mode effect analysis, and assign the occurrence number or the frequency number, then assign the severity number, and then assign the detectability number, and OSN. The multiplication, the product of these OSN is called as risk priority number in FMEA. So severity, occurrence, and detectability, 3 are the parameters. And in analysis, detectability is very important consideration. The high severity kind of cases will be rated very high as far as the FMEA score is concerned. High occurrence is also, rated very high, while low detectability will be rated very high. And the product of this is the product of RPN, risk priority number during failure mode effect analysis. So over here in this table, any number between 700 to 1000 is considered as high risk variables, and any number between 0 to 200 or 220 is considered as low risk, should be ignored, and any number between 220 and 700 can be called as intermediate risk variable, which can be, considered after brainstorming amongst the team. So failure mode effect analysis is applicable all over the industries, including the pharmaceutical industry, be it aeronautical, be it automobile, be it the chip industry or the pharmaceutical industry. So it requires brainstorming amongst the different team members, say quality assurance, quality control, say drug regulatory agency, drug regulatory division, IPR division, most importantly, pharmaceutical analysis division, product development division, and manufacturing division. They all sit together and decide upon which technique is to be used and which parameters are to be used and which are to be considered as the high risk, low risk, and medium risk parameters. So application of formulation by design is universal throughout the product development cycle. Not only it is at lab scale, pilot scale, exhibit scale, it goes beyond even post commercial launch, post launch, post marketing. So it even after approval, it goes around. So it leads to continuous improvement. So overall, we need to overall see a bird's eye view of the entire FPD as 4 steps: prioritization, then planning, because quality by design is quality by planning, then experimentation as per planning, and finally implementation. Prioritization, planning, experimentation, and implementation. Step 3, design guided experimentation and implementation. So in this case, the experimental designs are used, also called as statistical design, where the experiments are set up in as efficiently and precisely as possible. So it's a statistical design defining size and number of experiment units such that maximal information is obtained using minimal experimentation. So what are you observing? There is a melodious song, Hum Tere Bin Ab Reh Nahi Sakte. I think the movie, everybody, maybe younger scientists may be recalling, I think Ashiki. And there is the noise accompanying as well. So you are unable to enjoy the melody of the song because noise is also accompanying. Signal is the song, while the noise is a nuisance. So signal and nuisance, signal and noise are concurrent processes, concomitant processes. That's happening in day-to-day life as well. You tend to achieve success, but there are failures along with. You tend to achieve sukh, and there are dukhs along with. So every day does not is an amalgamation of only positivity, and positivity. So you have to adjust to both the kinds of situations. So in experimental design, you need to and you can decouple signal from noise. You cannot remove noise, but you can decouple. Signal को अलग कर सकते हैं, noise को अलग कर सकते हैं, and this way you can possibly maximize signal to noise ratio. That's what is needed. In pharmaceutical analysis, those who are experts in pharmaceutical analysis, those who are aware in pharmaceutical analysis also, it's required that signal to noise ratio needs to be maximized. So is done in case of the experimental design. Signal to noise ratio needs to be maximized. That is the beauty of the experimental design. Having decided upon the implementation of experimental design, we need to know about the power of experimental design, which is, again, time does not permit me, so which is based upon the alpha and beta power, or also called as type 1 error and type 2 error. So power is the effect of a factor. So to explain it better, I there is a golfer with a golf ball. Again, it golfer strikes the ball, you know, and the ball is struck. Ball is somewhere. Now he goes out searching for the ball, and the ball can be searched in the grass depending upon the diameter of the ball and the thickness of the grass. So if there could be, say, many cases, but these are the three exemplary cases. If the thickness of the grass, which is noise, which is preventing the visibility of a ball. The ball is the signal. The grass is the noise. So if the noise that the grass height is double than that of the ball diameter, as is its case, delta upon sigma. So 1 by half, the visibility of ball being spotted is just 15% from the top. But if it is equal, the noise and the signal is equal, the visibility of the ball increases from 15 to 45%. But if the signal to noise ratio is 2, means the grass is well mowed, and it's the height of the grass is one half of the diameter of the ball, the chances of it being spotted by the golfer is 96%. So this exemplifies the signal to noise ratio, which is a characteristic of the experimental design. So experimental designs are basically of two kinds. I have already discussed factor screening, which helps in the prioritization, and response surface designs, that's optimization designs. I'll discuss about these. The important experimental designs, time does not permit me to go into the details of any experimental design, but I'll just name to make you more conversant. Factorial design, central composite design, box Mencken design, simplex mixture design, or to be more precise, optimal design, fractional factorial design, Taguchi design, and Plackett-Berman design. These are the most common, most prominent designs. While the former, factorial, central composite, box Mencken, simplex, they are used for response surfaces, that is optimization. The later ones, that's Plackett-Berman, Taguchi design, fraction factorial, optimal designs, they are preferred for screening designs. While there are 2-3 designs which can be used for both optimal, fractional factorial, Taguchi design. But my dear ladies and gentlemen, let me share. So we need to have a common language between us to understand each other. So maybe English, maybe Hindi, or maybe any other language which is common between us. If you start speaking Tamil and I start speaking French, we some of us may not be able to communicate properly. So we need to have a common language, one common language. Purpose is served, but English would be better over here because there is enough vocabulary available, scientific vocabulary available in English to explain the scientific minutiae, the technical minutiae. So similarly, you need to have one design to experimental design to complete the task, but there could be advantages and disadvantages of each of these designs to come to the rescue. So this is how they are depicted: full factorial, fractional factorial, central composite, box Mencken, D-optimal, and Taguchi design. So time does not permit me to go into the details, but just a moment. If you look around, number of factors, influential factors, as they increase, how many number of experimental runs are increasing? So when there are 2, 4 using factorial designs, when there are 7, 128 studies are required. So those who are expert scientists, they will be knowing how voluminous this exercise is, just for optimization of the drug delivery systems, we may be requiring 128 experimental studies. Say for example, while preparing liposomes, you may be requiring different phospholipids, different cholesterol ratios, and different adjectivators, etc. Uh, so many 128 combinations are required, and then only we can embark upon the selection of the optimum formulation. ये तो अगर इंडस्ट्री में ऐसा करेंगे तो आपका मालिक, हमारा मालिक जो है इंडस्ट्री वाला, वो तो गश खा के गिर जाएगा यार, कि हर स्टडी में 128 करने. So minimum the number of experiments, more is the affordability, less are the number of experimental runs, and more is the affordability. So that's why many times fractional factorial design or central composite designs are preferred. So I am quite fascinated and satisfied with central composite designs. Say, for example, 4 factorial design, 1, 2, 3, 4, 5 central designs, and 4 axial points, total 13 points. Most of the, he is the father of experimental designs. So he used to call most designs are simple. I'm not able to discuss the details of merits of all these factorial, fractional factorial, Plackett, Budman, star. So central composite, there are advantages, disadvantages. Again, if you are interested, I can send you the papers where all these have been discussed in details, written by us, all papers written by us, where all these have been discussed. Step 4, modelization and validation of design and control spaces. So once we have discussed upon the experimental design application, so now we need to analyze that data, which is called as modelization, and we need to validate, and we need to embark upon design and control spaces. And for that, there are optimum search methods, which are graphical or mathematical or artificial neural network related approaches. Nothing to worry upon. We have to be aware, and we have to be implementing the apt usage of the computer software for the same. But you have to act as the master. Computer software would act as the servant. So in graphical techniques, brute force overlay, brute force is less common. Overlay plot is most common in graphical technique. And in mathematical, desirability function or numerical desirability function is considered to be very, very popular. Then there are some diagnostic plots. Again, this is computer generated diagnostic plots, which help you to diagnose which model is fit, and what are the problems, and what are the advantages of choosing a particular model. Say, for example, it tends to look into the residuals. Residuals means observed values minus predicted values. What you predicted on the basis of the model and what has been observed. The difference is called as residuals. So if the residuals, when plotted versus the original data, are quite regulated and quite randomized, as is the case in A, this is ideal situation. But if they are regulated but form a particular pattern, as this is convergent, this is having some kind of pattern, this is having U-shaped pattern D, and this is expanded at the peak in C. So this is also called a double bow. All these patterns show some kind of fishy processing. जैसे CID में कहते हैं कुछ तो गड़बड़ है दया, कुछ तो, so something is fishy, कुछ तो गड़बड़ है. So you need to work on the scientific basis. Yes, if the residuals are giving some kind of appearance. So this is providing you with the product and process understanding. Okay, and then this is another example of the models, use of models. Say, for example, this red experiment, you know, and this lead at the bottom has to reach at the top, which is the optimum, and you adopt a linear first order model like fractional factorial design, like Plackett-Burman design, you know, and you can reach close, but you cannot hit the bullseye. That's the optimum formulation. So if by reaching close, you adopt again the first order model, that Plackett-Burman design or fractional factorial design, you will miss the optimum formulation. But if by reaching close, you adopt the second order model, that is a central composite design, box Benken design, or simplex design, then you will hit the bullseye. It's just like, for reaching the Sharda, university, you may be adopting two-pronged approaches. You may come by, you may land up in the New Delhi airport by air, and from airport you have to adopt a taxi, and you have to come via road to Sharda University. Two-pronged strategies. एंड इफ यू विश के मैं शारदा यूनिवर्सिटी के ऊपर ही लैंड करूं, नॉट प्रैग्मेटिक. इफ यू विश के आप बॉम्बे से शारदा यूनिवर्सिटी बाय रोड ही आएंगे, अगेन इट्स नॉट कॉल्ड प्रैग्मेटिक. So you have to adopt many a time two-pronged strategy. First order model to reach close, and then second order model to reach at the optimum. So I have already shared explorable space is Noida, knowledge space is Sharda University, design space is the School of Pharmacy, and control space may be the conference venue. So you tend to remain within the design space. Design space has the regulatory importance, and within that design space you can move within control space. जैसे इंडिया में कहते हैं ना, थोड़े में गुजारा होता है. हमारे पास पैसा है पर हम थोड़े में गुजारा कर रहे हैं. So if you are going little beyond, you still remain within the design space. To make my point across, say if you have a farmhouse of your own, I wish everybody has got. So you tend to remain within the house. But once in blue moon, during a sunny day, you tend to go out and enjoy the green pastures around your house. And still you are remaining within your own house. Nobody can call that you have trespassed. But if you trespass beyond, anybody can say, gentlemen, you have now trespassed the boundaries of your design space. You are going beyond that, so which is not allowed. So you have to remain within your own house, ideally within that house, or you can go out and remain within the green pastures and still remain within your own precincts. And design spaces, we have to use appropriate tools to search for. And step 5, pilot plant scale-up, production and commercialization, which is carried out in industry. So pilot plant, lab pilot plant design space, same type of equipment is to be used, and commercial scale maybe wahi use hota hai. And tightening of operating space leads you to control space. Normally, you tend to move within the control space. 3 types of design space: lab scale, pilot space, and commercial scale. And normally validation is carried out in commercial scale, but it's very expensive. You carry out at lab scale and validate at commercial scale. And QTPP is the target, and it is within the design space. QBD is the vehicle on which you have to carry out. And there are software I had been talking about, which can come to your rescue. So Geometrics, and here is the best formulation, and you feel like rejoicing and celebrating. And but some people told that how to overcome the design space. So design space is essential, but it's not the single outcome of QBD exercise. It's the product and process understanding through science and risk-based QBD paradigms. I'm compiling in 5-7 minutes. So any fool can understand. The point is to understand, and any fool can know. The point is to understand. Einstein used to tell. So to make my point
He's going
across,
to pick the pink one. He's going to order samosas.
Samosa.
I know your heart only too well, so I give Kellogg's heart-to-heart oats every day.
so I bypass the second video. So to make your point across, the lady was understanding her husband. So that's why she could predict that he's going to now select pink tie. He's going to order for samosas, and when he's relaxed, he's going to put an arm around her. So because she understood, she could predict the next move. That's what FDA wants. That pharmaceutical scientists should understand the product and process and should be able to predict. That's why, there will be hardly any errors during pharmaceutical manufacturing, and the patients will not suffer. So where there is understanding, there is predictability. Where there is predictability, you can unearth the cause behind the problem. And so prior knowledge, prior publication, prior experience, prior patents leads to prior understanding. So how can product and process understanding, increase? I'll have to cut it short. Statistical quality control charts, chemometry, principal component analysis, and drug release kinetic modeling, various models, which can give you an inkling whether it's diffusion control release or erosion control release or swelling control release or biodegradable system or bulk erodible system or surface erodible system. In vitro, in vivo correlations can give you enough of information whether the in vitro studies are simulating the in vivo performance or not. And there are various IVAVC models which can be implemented. There are software, again, to come to your rescue. And then PAT tools, process analytical technology tools, are very, very important. Again, time does not permit me. PAT goes in tandem with QBD, and there are non-destructive tools like FTIR and terahertz spectroscopy. And design, analysis, and control is the basis of PAT. PAT banks upon QBD. And its QBD is very much getting implemented in novel drug delivery and nanostructure systems. This is out of one of the review articles which we published in December in 2021, which contains data till 2020. You can see the exponential usage of QBD in the development of nanostructure systems. And this is applicable to all kinds of drug delivery systems, be it nanovascular systems, nanoemulsions, nanocrystals, and all kinds of designs have been adopted. BBD, CCD, and FD are most common. And we hear a lot of talk on continuous manufacturing, where batch manufacturing will be replaced with continuous manufacturing. And this is being promulgated by US FDA time and again, where PAT and QBD both will be implemented. So traditional manufacturing and QBD would lead to continuous manufacturing through blue sky vision and other kinds of paradigms. So exploring continuous manufacturing, USP has been emphasizing. So I bypass.
Manufacturers of these.
I bypass. And QBD for efficient pharma development, is it an option, coercion, or benediction, blessing? I find this as benediction. So pharma excellence and regulatory compliance both are, coming from the single usage of QBD. So it's applicable to API development, analytical development, formulation development, dissolution testing, manufacturing process, bioequivalence study, stability testing. And it's applicable for translational research as well, where science gets converted to technology from bench to bedside stories. And pharma quality by design, when the whole world is adopting, when the whole world needs, the developed world needs, where are we? Are we still thinking? Are we still in the denial? That's the scenario. Let me share So our contribution, as already Dr. Ankita has pointed out, so we have had 9 consultancy projects, 18 pharma companies we have trained for beyond the Indian precincts in Taiwan, in Canada, in Dubai and Bangladesh, and 14 in India. And it's 66 H-index and 16,000 citations and 10 patents and 3 tech transfers, in collaboration with my colleague, Professor O.P. Katare. And this is how we are working in tandem with the pharmaceutical industry in India and beyond. And these are the 2 technologies. Third, I have not included. These are the highly cited and highly used articles on the FBD and QBD, and 10-volume book series on nanobiomedicine. Mostly, I had the opportunity of working on nanostructured systems as the founder coordinator of UGC Center for Excellence in Nanobiomedicine for 10 years. So this is the CEO of Statis company, the company that markets and develops design experts. So they sought our help, and later on they awarded the Statis Pharmaceuticals product performance. And till today, 2013 until 24, we are getting greatest supply of the perpetual and single user licenses. These are some FDA reviewers: Dr. Vilayat Saeed, Dr. Lane Christensen, and Dr. Albinus D'Souza, with me and Professor Saranjit from NIPER. And Dr. Ejaz Hussain, he who implemented QBD while he was in FDA. So he was there with us in one conference. And so some awards, international awards at China in 2019, and my students getting award to Dr. Rishi Kapil, Dr. Kesar Raza at AAPS, American Association of Pharmaceutical Sciences QBD Award, Dr. Sarwar Beg, Bexi Research Foundation Award, and Dr. Rajneet, getting QBD Pharma Development Award, and our UIPS getting second rank and from the Ministry of Human Resource Development in 2016, and we continue to remain in top 2 or 3 in research parameters. We tend to emulate Professor Kishan Singh Ji, whom we lost in 2021, and he only Padma Shri awarded in the pharmaceutical sciences. So but one point of concern, which Mr. Narayana Murthy is making, that death of 66 children in Zambia. This is because of the loss of integrity. So we need to work on quality as well as integrity. Attitude determines altitude. We need to adopt our attitude, and then the altitude will automatically be attained. Stance and stand matter a lot. We need to adopt this stand. Always stay positive. Chalta hai attitude needs to be, converted to chalna hai attitude. Jugaad technology has to be converted to Juran technology. We need to fly at the winged pace and stand apart in the world of mediocres. Then the day is not very far when our mother India would stand prominently on the world map, and together everyone achieves more. This was my team in 2021, along with some of the colleagues, Professor Katare and other colleagues. Thank you. This is Chitkara University, and this is Punjab University. Thank you so much for your patient listening. I'm available for any.
Thank you so much, sir, for such an informative and interesting deliberation. Especially the examples which you quoted were really commendable. I could not even, for a second, I could not keep my eye off your deliberations, sir. Thank you so much.
So nice.
I hope the participants have learned a lot from this about quality by design.
Yeah.
Thank you so much, sir. I also
the participants, those who are inquisitive, the purpose will be served only if their queries are solved. So they can route their queries through you people to me, and at my earliest convenience, I will answer to all those queries, try to answer all those queries to them, of course, through you only. Okay.
Sure, sir, we'll look into this. So, with this, we come to the end of day one of our 5-day international FDP. I would like to thank Professor Ranjit Singh sir and Professor Bhupinder Singh Bhoop sir to grace this occasion by their presence and for sparing their invaluable time with us. I would also like to thank all the participants which have joined us online through this FDP. And for the information, we'll be resuming tomorrow at 12 o'clock with our speaker, Professor Odeku from Nigeria. And the link will be shared on the group which has been created on the WhatsApp. So thank you so much and have a very nice day.